In this video, we will discuss about DNA methylation. So, why we are discussing about DNA methylation as is it and as this is also one of the cause for cancer. So, there are two kinds of DNA methylation that can occur. One is during embryonic state, that is during differentiation from embryonic states to a committed cell, and another is during committed cells. So one that occurs during embryonic state is being done by DNMT 3A 3B. What is DNMT? DNMT is DNA methyl transferase. 3A 3B are the different form of DNMT. So there are two kinds of there are two ways by which DNA methylation uh, can inhibit the expression of genes, thereby alter the epigenetic changes. And previously it is understood, or it is well known uh, well-known acceptable thing that cancer occurs because of genetic changes, that is changes in the DNA. However, recently people have identified that not only the genetic mutations or is responsible, but but the epigenetic changes, uh, that is the change in the expression of genes, is also responsible for uh, generation of cancers or initiation of cancers. So let us understand the first part. What does this first part, A panel of the figure shows that if this thing happens, its final result will be the inhibition of transcription. That is, a particular gene will be inhibited from transcription, and that gene could play an important role in the cell cycle. So how this happens is that the uh, this is HDAC, that is histone diacetylase complex. The, it might bind to a particular section of the gene, and this is uh, this is methyl methyl binding domain protein. This is methyl binding domain protein, and this HDAC and MBDP will interact together. That is, it is shown by this interaction. It will interact together to finally bind to a particular gene, thereby preventing RNA polymerase to convert this gene into the gene into the mRNA. That is the process of translation, and after that, further from RNA to polypeptide chain formation is known as the process of transcription. So if this initial step, if this MBDP and HDAC binds on the section of gene, it will prevent RNA polymerase 2, thereby preventing the final transcription of this gene, thus affecting the normal cell cycles of the cell. And this might happen to some of the important genes that are essential for the healthy cell cycle. And the second thing that it will happen is the methylation initiate. How the methylation initiated in the regular cell. So this is the CPG island. What is CPG island? CPG island is normally called as the stretch of DNA. As we know that DNA has four bases: A, T, C, G. Adenine, uh, thionine, guanine, cytosine. So, and in this CPG island is the stretch of DNA which has a long portion of GC nucleotide. For a general idea, we can say that if there is a 200 base pair, 50% of this nucleotides combination should be GC. So what happens is that this DNMT1, as we know that DNMT 3A and 3B will form methylation during embryogenic stage, and DNMT is uh, DNA methyltransferase. DNA methyltransferase 1 will bind to one of the section of the gene, or we can say that in one of the part of the CPG island, and the closed or the pink color bubble represent the methylated CPG part, and the open circle represent non-methylated CPG part, and it will then bind to UHRF. UHRF is ubiquitin like protein that has ring structure and another structure that I'm not able to recall it right now, and they both can form a methylation structure with the help of at the position that is H3K9, that is histidine at third position and lysine at 9 position, and these are this location of the amino acids or the residues are methylated. So when this combination forms, it will initiate the methyl formation of this CPG island, uh, which will prevent the transcription and then translation process of this kind of gene. So both of these will affect the epigenetic or cause the epigenetic change, that is change in the gene expression, which might cause uh, changes in the cell, and that might even lead to cancer. It is not 100% that uh, this methylated DNA methylation might lead to cancer. However, if this DNA methylation occurs on the gene that are essential, then it might initiate the cancer, or propagate or accelerate the cancer formation. And this strategy has been found out in one type of cancers, such as breast cancers, where if DNA methylation has been observed in the those cancerous cells, and there is a strategy, or we can say that uh, uh, people have identified a way to reverse this DNA methylation. So if DNA methylation is found in the cancerous cell, if and they are being treated with that process to undo this DNA methylation, then people have observed that the survival rate of such persons has increased. So, so this kind of this information about DNA methylation has helped to has helped the science to find out a cure that can increase the life of the persons who are suffering with a cancer. So that's all for this video. Thank you for your time.